from 2 speakers and then hold a panel discussion before the lunch break. Dr. Connie Ruzicka is a laboratory branch chief in the Division of Pharmaceutical Analysis within OPQ's Office of Testing and Research. She plans, directs, and manages the work of laboratory personnel performing testing and modern research that promotes the use of advanced techniques to characterize drug products. And Neil Stiber is the Associate Director for Science and Communication in OPQ's Office of Quality Surveillance. During 8 years at Cder, he has innovated risk-based approaches, provided collaborative program leadership, and engaged stakeholders to advance pharmaceutical quality. Let's turn our attention over.
Thank you for that nice introduction. My name is Connie Ruzicka, and I'm a laboratory branch chief in the Division of Pharmaceutical Analysis within the Office of Testing and Research. And today I'm going to talk to you about how analytical results can be used to assess the quality of drug products in order to keep safe products in the marketplace for consumers. The learning objective of this presentation is to describe how science drives regulatory actions to protect consumers from unsafe products utilizing the quality assessment of hand sanitizers as a case study. An overview of this talk is that we're first going to discuss the importance of hand hygiene, discuss the agency's temporary hand sanitizer guidance and how this policy was used to influence laboratory testing, discuss the development and implementation of analytical methods for quality assessment of hand sanitizers, and discuss how laboratory results can be used as evidence in enforcement actions. So to get started, we're first going to talk about hand hygiene. Hand hygiene is important to mitigate the spread of COVID-19, and the CDC recommends use of alcohol-based hand sanitizers when soap and water are not available. Throughout this presentation, you will see that we will refer to alcohol-based hand sanitizers as ABHS. In addition to hand sanitizer products containing alcohol, there are also products in the marketplace that have benzalkonium chloride as an active ingredient. For the presentation today, we will only focus on alcohol-based hand sanitizers. Hand sanitizers are over-the-counter antiseptic drug products frequently used by consumers. They're used to prevent the spread of infections and to decrease the risk of getting sick. At the onset of the pandemic, there was difficulty in accessing alcohol-based hand sanitizer products due to an increase in demand. In response, FDA issued a policy for testing alcohol for methanol and a guidance to communicate its temporary policy for the preparation of alcohol-based hand sanitizers. This here, as you can see, is an example or as a cover of the temporary policy that was issued by the FDA during the COVID-19 public health emergency. The temporary hand sanitizer guidance was for industry, and it was intended for entities not previously licensed or registered drug manufacturers. As part of the temporary policy, it did identify that the CDC recommends alcohol-based hand sanitizer products to contain at least 60% ethanol to be efficacious. In addition, it also identified some additional considerations for the ingredients used in hand sanitizers and provided general formulations that should be used in producing hand sanitizers during the public health emergency. Part of these considerations for the ingredients was that one, the alcohol meet USP or FCC grades, and also that alcohols are screened for potential or harmful impurities. One such impurity is methanol. There have been cases where methanol has been substituted into hand sanitizer products. Methanol is toxic, and in some cases, hand sanitizers with methanol were ingested and did cause severe adverse events, including blindness and death. Recently, a Federal Register notice has been announced to withdraw the hand sanitizer temporary policies at the end of the year. So now that we have a background and we know the importance of hand sanitizers and with the temporary guidance and its use of the temporary guidance, we have to determine how we're going to move about using laboratory assessment of these products and how we can ensure that safe products are in the marketplace. So the goal was to develop a testing protocol that we could evaluate the quality of alcohol-based hand sanitizer products using the guidelines provided in the temporary policy. So this method had to be comprehensive. We wanted to be able to first identify the alcohol used in the hand sanitizer product. Approved alcohols for hand sanitizers include ethanol and isopropanol. But we also wanted to ensure that no alcohol substitution was taking place. So we wanted to be able to identify additional alcohols such as methanol or 1-propanol that have been used in hand sanitizer products in addition to the 2 approved ingredients. In addition to identification, we also wanted to determine the percent alcohol content or the assay of the product. We wanted to ensure that the appropriate amount of alcohol was being used and that the product had efficacy based on the CDC guidelines. Lastly, we wanted to screen for impurities. As we mentioned, one of the considerations was to use alcohols having USP or FCC grade. Fuel-grade alcohols can have a higher content of impurities, and the impurities of interest for these products are shown in the table provided on the slide. For the top 4 impurities, methanol, benzene, acetaldehyde, and acetal, are known as our level 1 impurities. These are known as to be genotoxic as well as carcinogenic. They have much lower limits, as you can see, not more than 2 ppm for benzene and 630 ppm for methanol. Acetal and acetaldehyde have not more than 50 ppm limits. The other impurities are known as our level 2 impurities, and those have much higher impurity limit specifications. The first method that we developed was a rapid screening method for alcohol-based hand sanitizers. The method utilized spatially offset Raman spectroscopy, SORS, and it was able to screen samples through the container. One of the advantages of using a rapid spectroscopic screening method is that it can quickly screen products, and these methods were specifically used to identify alcohol substitution or samples with subpotency, 2 of the main characteristics we identified in the development of our method. Next, these types of methods can be used to increase throughput and prioritize samples for additional testing. Rapid screening tests can be done within minutes, and it allows us to identify violative products that will require additional testing to screen for impurities. Lastly, results used from the rapid screening can be used to provide up-to-date public notices. They can provide prompt regulatory actions, and it can help quickly remove harmful products from the marketplace. In developing the rapid screening method, or the SORS method, we first needed to build a custom hand sanitizer library for alcohol substitution and to assess the assay values. The method was developed with utilizing a variety of alcohols. As we mentioned, we wanted to look for not only the active alcohol ingredients such as ethanol and isopropanol, but we also wanted to look for those that could be used in substitution, which include 1-propanol as well as methanol. And you can see here the limit of detection table that is provided, that in many of the cases, the limit of detection in the hand sanitizer products can range between 2 and 4% volume to volume in hand sanitizer products. While these values or limits of detection are much higher than what we would like for hand sanitizer products, it is a good way to screen products and identify those products that are specifically well outside of the specifications. The benefit of using rapid screening is that samples can be analyzed in less than 25 seconds, and it results in a pass-fail results. As I previously mentioned, the method lacks sensitivity to detect impurities at levels specified in the guidance. So therefore, we had to develop additional analytical methods to further characterize the drug products. The spatially offset Raman rapid screening method has been used to screen over 200 hand sanitizer samples, and you can see here from the graph shown that we've tested close to 150 samples with ethanol as the active ingredient and over 50 samples with isopropanol as the active ingredient, and we've had less than 50 samples fail. When utilizing the rapid screening technique, many of the samples that are failing have often had alcohol that was identified in the product that differs from the label claim ingredient. For example, the label claim may say ethanol, and when testing the product was tested positive or was found to identify isopropanol as the active ingredient. We've also identified subpotent products that have less than the recommended alcohol content provided by the CDC. For example, we've had products that had less than 60% volume to volume of ethanol, in which the sample would not be within an efficacy range to help prevent any spread of disease. The drawback of using SORS is that it will require confirmatory testing. The method does not address all of the quality concerns outlined in the temporary hand sanitizer guidance. And as you can see here, the method that we've developed through SORS has been published, and we have a reference here that you can look up if you would like additional information. So the first challenge question I have for you is, what alcohol has been found in alcohol-based hand sanitizers that has caused a significant number of adverse events, including death after ingesting? Is it A, ethanol, B, isopropanol, C, methanol, or D, 1-propanol? If you selected C, methanol, you're correct. Now that we talked about utilizing the SORS method for rapid screening, we had to develop a method that had the ability to meet all of the criteria in the temporary hand sanitizer guidance. And our gold standard method is utilizing gas chromatography mass spectrometry. And the reason for that is that the samples analyzed, the alcohols as well as the impurities, are all volatile, which makes this a very good method. We were able to develop a single method for both assay and impurity determination. In addition, this technique has acceptable specificity and sensitivity for all active ingredients and impurities identified in the guidance. Another benefit is that the method can be applied to all formulations of alcohol-based hand sanitizers. This includes liquids, gels, foams, and wipes. And for this particular presentation, we are mainly focusing on liquid formulations, as that is what is presented in the temporary guidance. So for the Gcms testing protocol, as we mentioned, we first do identification of the alcohol. We screen for the active ingredient, ethanol or isopropanol, and also have the ability to look for alcohol substitution and include methanol or 1-propanol. We also can determine assay or alcohol content. Once we determine the percent volume to volume alcohol, we can compare that to the label claim to determine the specification if it meets the specification for label claim, which is 90 to 110%. Then we test for impurities, and we have several different routes for testing impurities. First, we have the semi-quantitative route for acetal and acetaldehyde, and why that is semi-quantitative we'll go into in the next few slides. We can also quantitatively determine methanol and benzene, and then we perform a limit test for the level 2 impurities, and as you can see, those were the ones with a much higher limit. In addition to the impurities quantitative screening, we also do spike and recovery, as hand sanitizers formulations can vary. Some have colorants or fragrances or even moisturizers, and we want to make sure that the results that we're obtaining for the impurities as well as for the alcohol content are not affected by the matrix. When we first began our testing alcohol-based hand sanitizers, we noticed that there were some irregularities in the spiked recovery for acetal and acetaldehyde that was observed during the analysis of several ethanol-based samples. As you can see here, this table represents, or this bar graph shows, the deviations that we've seen in yellow for acetaldehyde and acetal. What we noticed is that looking at the pH, we would see variability in the recovery for these samples. If we remember, the acceptable range for spike recovery is 80 to 120%. And we see much lower acetal recoveries for lower pH samples. And then we also seen higher recoveries for pH samples within 4.6 that were well above 140% for acetaldehyde. So this was very concerning. Looking into the literature and understanding the chemistry that occurs, we determined that in neutral and basic conditions, acetal is chemically stable, but in acidic conditions, it can hydrolyze into acetaldehyde and ethanol. So what we were hypothesizing is that the acetal in hand sanitizer products at lower pH was converting to acetaldehyde, and in that process could also generate ethanol. So based on the samples that were collected, there were over 265 ethanol-based samples that were collected during the surveillance study. So we tested the pH of all of those samples and identified that the samples had a range of 3.4 to 9.4, and that nearly 60% of the samples had a pH greater than had a pH less than 6, as you can see here from the pie chart on the slide. So now that we've gone over some of the methods that we use, I want to give you an example of one of the samples that was tested. And this sample is identified by a number here, and the active ingredient on the label claim was 80% volume to volume ethanol. We measured the pH of that sample, and it was determined to be 4.5. The assay value that was determined experimentally was 82.2%, and that was within 102.7% of the label claim, meeting the percent label claim specifications. When testing the impurities, you can see here that the sample for acetal and acetaldehyde failed with high levels of impurities. Remember that the interim limit for these specific impurities is not more than 50 ppm, and that during our analysis we detected 283 and close to 2,000 ppm for acetal. The sample had to undergo an extra dilution as it was outside the linear range that was determined and developed in our method. From what we can see here, that this was confirming our hypothesis that at low pH or in acidic samples that we would see an increase in acetal and acetaldehyde, and this sample specifically failed for those two impurities, and this trend has continued on with additional samples that we've tested. So my next challenge question for you is, what impurities are difficult to quantitate in alcohol-based hand sanitizer products with acidic pH or pH less than 6? Is it methanol and acetal, acetal and acetaldehyde, methanol and benzene, or acetal and acetone? It is acetal and acetaldehyde. So now that we've developed our methods, our rapid screening method and our GC method, we've been able to collect over 325 samples in our surveillance assignment, and 75% of those samples contain ethanol as the active ingredient. And in this slide here, you can see that we are still in progress of testing these samples. We still have a lot of samples pending. 47 and 79% between isopropanol and ethanol samples are pending. We've had 44% of the isopropanol samples pass and meet specifications, and while 10% of the ethanol samples have met specifications. What you'll notice here is that approximately 11% of the ethanol samples that we've tested so far have failed to meet specifications, and 9% of isopropanol samples have failed to meet specifications. Based on the samples that were determined to be violative and found to not meet specifications, we have outlined them here as to what is causing the violative results. You can see that over 75% of the samples are containing acetal or acetaldehyde, which is consistent with the pH studies that we performed, in which we assumed that a majority of samples at lower pH would have higher levels of acetal or acetaldehyde impurities. So now that we've demonstrated the use of the analytical methods, what are the results useful for? Well, the results can be reviewed by our Office of Compliance, and they can determine a variety of regulatory actions. For import samples, samples can be refused admittance. The agency can also put out internet and social media warnings. They can put out public notifications. There can be enforcement and recall information that's sent out. Since the beginning of testing of hand sanitizers, some firms have recalled poor quality products based on the laboratory results determined within OTR. More than 250 products are recommended by FDA on the do not use list. And here there is a link that can connect you to the do not use list. It's important that consumers go out to this list to identify if any of the hand sanitizer products that they're using have quality issues and are listed on the website. So in summary, FDA has tested alcohol-based hand sanitizer products for quality because it's a product under its regulations. FDA laboratories developed and validated analytical methods to assess the quality of alcohol-based hand sanitizer products using a rapid spectroscopic screening method such as SORS and a gas chromatography mass spectrometry method that has the ability to perform the identification of the active ingredient, quantitate the active ingredient, as well as screen for impurities that could be a result of use of wrong grade of alcohol. When testing alcohol-based hand sanitizers, there were serious safety concerns were identified from the laboratory results. First, there was contamination with potentially toxic ingredients, such as using methanol or 1-propanol in the product versus the approved alcohols as ethanol and isopropanol. Also, high levels of impurities are also being identified in these products. Impurity levels greater than those specified in the temporary hand sanitizer guidance. In addition, we've also identified subpotent or hand sanitizer samples that did not have enough active ingredient and are not within the efficacy range as identified by the CDC. We've also identified products with false labeling where they would identify one alcohol and contain a second, or have misleading or unproven claims. So based on the presentation provided today, there are a variety of resources that you can use. You can go to the website here in order to find, look at the hand sanitizer temporary guidance. There is the withdrawal notice. There's also a link to the hand sanitizers do not use list, as well as a reference to the publication that summarizes our SOARS method. And I would like to take time to acknowledge all those that have helped with this work. This is the study has been extremely intense and ongoing due to the pandemic, and it takes a village in order to help screen these products, ensure that safe hand sanitizers are available in the marketplace. So with that, I would like to take any questions. So I'd like to leave you with one closing thought. Laboratory science drives regulatory action to protect consumers from unsafe products. Even over-the-counter products like hand sanitizers can be susceptible to quality deficiencies. It's important to utilize analytical methods that can identify these deficiencies and ensure that safe and effective products are available to consumers in the marketplace. Thank you. Thank you for your attention. And if you have additional questions, please feel free to email me those. My contact information was on the previous slide.
Good afternoon. My name is Neil Stiber, and I'm the Associate Director for Science and Communication in OPQ's Office of Quality Surveillance. I'm pleased to speak with you this afternoon about the report on the state of pharmaceutical quality, known as the Rsvq. There are three main learning objectives for today's talk. Number one, I would like for you to understand the purpose of the report on the state of pharmaceutical quality, the Rsvq. Secondly, I would like for you to be able to describe at least one impact of the COVID-19 public health emergency on drug quality surveillance. And third, I would like for you to know the approximate distribution of sites in the Cder site catalog with respect to whether or not they are listed in an approved application. I'll start with some background about the Rsvq and my sub-office within OPQ, the Office of Quality Surveillance. Then we'll speak about the Rsvq Fiscal Year 2020 report, the most recent Rsvq report. We'll close by talking about OPQ's commitment to quality and talk a little bit about the Rsvq Fiscal Year 2021 report, which is in development. I want to start with a little background about my sub-office within OPQ, the Office of Quality Surveillance. OQS continuously monitors and provides the state of quality for all regulated sites and products. The availability of quality medicines is assured through OQS actions that include signal detection, data analysis, review of the state of quality, and proactive stakeholder engagement. OQS created the annual report on the state of pharmaceutical quality to support OPQ's mission and vision and to provide useful information for the public. And we hope that this report is not only useful to us internally, but is also insightful for you outside of FDA. Now a little background on the Rsvq itself. The report on the state of pharmaceutical quality characterizes the state of pharmaceutical quality for human drugs legally marketed in the U.S. for each fiscal year. It identifies indicators or elements of quality based on analyses of available FDA data for site compliance, post-market reporting, and product testing. In doing so, We hope that it will provide insights and trends that can be inferred about site quality and product quality. These can provide lessons that are useful to FDA and useful to the public. The Rsvq has been published for 3 years, and each of these reports are available for you to read on the web. Now let's talk about the fiscal year 2020 Rspq. It has several sections, including those which I will speak about today on manufacturing site demographics, impacts of COVID-19, manufacturing site compliance, and drug product quality. The report also provides findings including innovative data analyses, for example, cluster analysis, statistically significant trending, and machine learning models. Let's start with manufacturing site demographics according to the industry sectors. Sites in the Cder site catalog are characterized through a hierarchy of industry sectors. This hierarchy starts with BLAs. 10% of all sites are listed in at least one biologics license application. Of the remaining sites that are listed in approved FDA applications, 12% of sites are listed in at least one new drug application, but not in any ANDAs or BLAs. 14% of sites are listed in at least one abbreviated new drug application, but not in any NDAs or BLAs. In addition, there are 26% of all sites that are listed in both ANDAs and NDAs. The remaining 38% of sites are not listed in any approved FDA applications. These sites include OTC monograph sites, unapproved product sites, and homeopathic sites. Overall, 62% of all catalog sites are listed in application products, and 38% of all catalog sites manufacture exclusively non-application products. That's an important number to keep in mind. Now let's talk about some of the impacts of COVID-19. From a quality surveillance perspective, one of the most important impacts of COVID-19 was the postponement of non-mission critical inspections. In response to the postponement of these inspections, FDA applied and innovated strategies from existing authorities to assess regulatory submissions that would typically have needed pre-approval inspections or pre-license inspections. These strategies included reviewing surveillance history, requesting records and information under Section 704A4 of the Food, Drug, and Cosmetic Act, and using inspection reports that were obtained through MRA, Mutual Recognition Agreement partners, including third country reports. Using these strategies, Cder completed facility assessments to meet user fee dates over 90% of the time and reduced the need for PAIs and PLIs. Another important impact of COVID-19 was for hand sanitizer products. Starting in March 2020, FDA issued guidance detailing temporary policy for alcohol-based hand sanitizer products. This policy allowed firms to register and manufacture hand sanitizer products without being subject to routine surveillance inspections. As a result, more than 5,000 firms, including distilleries and other industrial manufacturers, registered. After FDA outreach to confirm the registrants and their products, approximately 2/3 of these firms deregistered from the Cder site catalog. And currently, there are approximately 1,500 newly registered hand sanitizer manufacturers. To provide surveillance oversight on these firms, FDA conducted sampling and testing of imported and domestic hand sanitizer products. This provided an estimate of the quality of newly registered firms. FDA also assigned for-cause sampling assignments based on complaints that it received. FDA established a should-not-use list to identify manufacturers that were considered higher risk, and also established a countrywide import alert for hand sanitizers from Mexico. This chart shows the trending of hand sanitizer complaints during fiscal year 2020. As you can see, following the temporary guidance, there was a significant increase in the number of complaints, which peaked during the summer of 2020. Now let's speak about manufacturing site compliance. Due to COVID-19, the postponement of all non-mission critical inspections began in March 2020. Nevertheless, FDA still performed 562 drug quality assurance inspections in fiscal year 2020. Most of these were before the public health emergency. In addition, the mutual recognition agreement was used to assess 183 sites. And so in total, for fiscal year 2020, there were only 745 sites, 18% of the Cder site catalog, that received surveillance inspection coverage. By comparison, during fiscal year 19, there were 1,367 sites, 32% of the Cder site catalog, that received surveillance inspection coverage. Let's talk about the warning letters as a function of industry sectors. Current good manufacturing practices violations that are observed either through an inspection, violative evidence collected from a record request, or from failing analytical sampling and testing may result in a regulatory action, including warning letters. As you can see in the chart on the right, the number of warning letters issued in fiscal year 2020 was slightly lower than in fiscal years 18 or 19, but it was still more than 4 times higher than in fiscal year 2015. As in past years, most warning letters were issued to sites manufacturing exclusively non-application products. This no-app sector, as you can see in the blue parts of the bars on the right, constitutes a majority of recent warning letters. It should be noted, as we said previously, that only 38% of the Cder site catalog is from this no-app sector. Now let's talk about drug product quality from a post-market perspective. The FDA receives feedback from industry, healthcare providers, patients, and consumers on product quality via several types of product quality defect reports. These PQD reports include field alert reports, MedWatch reports, biological product deviation reports, and consumer complaints. Let's look a little bit more into these product quality defects. The FDA collects PQD reports, assesses the defect severity, and looks for trends and ways to mitigate issues. During fiscal year 2020, FDA received thousands of PQD reports, including nearly 12,000 MedWatch reports, more than 4,000 FARs, and several hundred BPDRs and consumer complaints. As can be seen in the pie chart on the right, these PQD reports can be grouped into 20 defect categories. And as this chart shows, over the past 5 years, there are 3 categories that have constituted more than 60% of all PQDs. These 3 categories are product quality questioned, device issues, and packaging issues. Now let's speak about recalls. In recent years, major recalls have been associated with microbial or chemical contamination and impurities. This suggests a focus area for industry to improve quality and for FDA to increase oversight. As an example, in fiscal year 2017, microbial contamination caused recall of antibacterial products. In fiscal years 18 and 19, nitrosamine impurities caused recalls of several cardiovascular products. And most recently, in fiscal year 2020, nitrosamine impurities caused the recalls of gastrointestinal products. I want to talk with you a little bit about our plans for the Rsvq report for fiscal year 2021. This is a report that's under development, and we're currently identifying relevant questions that will be of value to the public and the FDA. It's likely that it will reflect the unique nature of fiscal year 2021 with the public health emergency and COVID-19. There were postponed inspections, and FDA continued to use alternate tools for quality surveillance. For example, Section 704A4 of the Food, Drug, and Cosmetic Act. It's likely that the Rsvq fiscal year 2021 report will also have discussion on the quality of hand sanitizer products, as was part of the 2020 report. As a summary of today's talk, I would like to emphasize OPQ's commitment to quality. The report on the state of pharmaceutical quality annual report summarizes various measures of the pharmaceutical manufacturing industry's ability to deliver quality drug products to U.S. patients and consumers. In doing so, it enables the public to understand the pharmaceutical industry and how FDA acts proactively to address potential pharmaceutical quality issues before patients and consumers are impacted. And now I have several challenge questions to share with you. Number one, true or false? FDA's temporary policy for alcohol-based hand sanitizer manufacturers allows firms to manufacture hand sanitizer products without being subject to good manufacturing practices. The answer is false. All firms are subject to good manufacturing practices. The temporary policy allows firms to manufacture without being subject to routine surveillance inspections. Challenge question number two. Which of the following is not a type of product quality defect report that is used by FDA? A, MedWatch reports. B, eBay customer ratings. C, field alert reports. D, biological product defect reports. Or E, consumer complaints. I hope you all recognize that eBay customer ratings are not used by FDA as a type of product quality defect report. And now challenge question three. When did consumer complaints about hand sanitizer products peak? A, May and June 2020. B, July and August 2020. C, January and February 2021. Or D, June and July 2021. The answer is July and August 2020. That is when there was a peak of complaints about hand sanitizer products. I want to close with this thought. Based on everything we've learned and we've shared with you today about the report on the state of pharmaceutical quality, I think we can say that U.S. patients and consumers deserve confidence in their next dose of medicine. Thank you very much for your time and attention. I'd be happy to answer your questions.
Thank you for that great presentation, and welcome to our second question and answer panel. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Looks like we have some questions coming in right now. And the first group of questions coming in is for Dr. Connie Ruzicka. And here's the first question for Dr. Ruzicka. You mentioned that a Federal Register notice has been issued for the withdrawal of the temporary hand sanitizer guidances. Can you please provide more details pertaining the withdrawal?
Thanks, Ray, for that question. That's a very good question. The temporary hand sanitizer guidance withdrawal notice went out just a couple weeks ago, and as part of that, it states that firms manufacturing hand sanitizers under these guidances can continue to produce manufacturers through December 31st of this year. They have the ability to distribute those products through March 31st of this year. So After that, the hand sanitizer firms manufacturing products under the hand sanitizer guidance can no longer do that as the guidances will be withdrawn. But those are the dates provided in which they can continue to manufacture through the end of the year and continue to distribute through March. And back to you, Ray.
Thank you for responding to that question. We do have another question for Dr. Ruzicka, and here's the question. Alcohol substitution has been observed in hand sanitizer products, but the pie chart on slide 16 does not list or provide a percentage for hand sanitizer products found to be volatile due to alcohol substitution. Why is this?
That's a very good question, Ray. So the presentation today really focused on the surveillance data from domestic hand sanitizers. And currently the samples being tested within the Office of Testing and Research has not found any substitution within the domestic market. Most of the substitution products that have been identified through testing have been found through imports. Back to you, Ray.
Thank you for responding to that question. We have another question for Dr. Ruzicka. Here's the question. Where do the impurities listed in the hand sanitizer guidance originate?
Thank you. Thank you for that question. So impurities from the hand sanitizer guidance really were focused on the potential of using fuel grade alcohol in the production of hand sanitizers instead of using USP or FCC grade. So in fuel grade alcohol, there is the potential for higher level of these impurities. And we wanted to ensure that these toxic and genotoxic impurities were not present in the hand sanitizers produced under the temporary guidances or policies. So we recommended the use of FCC and USP grade. Thank you for that question. Back to you, Ray.
Thank you for responding to that question, Dr. Ruzicka. We do have another question for you, and here's the question. How can someone be sure the hand sanitizer product they are using is safe?
That is an excellent question. So we do post all of our information on regarding the safety of hand sanitizers on the FDA website. So we recommend that folks go out to the do not use list. That was referenced in my presentation. It was also referenced in Neil's presentation. And if you do a Google search on the website for FDA hand sanitizer do not use list, you can find a list of products that were tested and that the FDA does recommend to not use. Thank you, and back to you, Ray.
Thank you for responding to that group of questions. We're going to move over to Dr. Neil Stiber. And we have a group of questions coming in. Here's the first question for Dr. Stiber. How can one access the Cder site catalog?
Hi, Ray and everyone. Good afternoon. Thanks for that question. The Cder site catalog is an internal database that FDA uses to track the sites who are registered to manufacture drug products. So that's an internal information source for us. What I think is special about the report on the state of pharmaceutical quality is that we've provided a lot of summary statistics from that site catalog that hopefully better characterize the state of quality and are helpful to the public. Back to you, Ray.
Thank you for responding to that question. We've got a few more questions for Dr. Stiber, and here's the next question. How do the inspection and mutual alliance numbers look for fiscal year 21 compared to fiscal year 20?
Great question. Well, what I'd like to say about that is that I don't have specific numbers to tell you right now, but certainly the number of inspections that FDA has been able to conduct, as well as the number of inspections that our partners in Europe were able to conduct, were greatly impacted, of course, by the public health emergency. And so the numbers for fiscal year 21 are going to be significantly lower than for fiscal year 20. For fiscal year 20, more than half of the year, about half of the year, was before the public health emergency. And so that's something to expect for those numbers. Thank you, Ray.
Thank you for responding to that question. The next question for Dr. Stiber. Dr. Stiber, you showed only a few hundred BPDRs. Does this reflect all BPDRs or only those sent to Cder?
Yeah, that's a great question. Thanks for the opportunity to make that clarification. The numbers that we reported were for BPDRs of Cder-regulated products. And of course, there are also Stiber regulated products that would also have BPDRs, and we didn't present on those. So thanks for that opportunity for the clarification.
Thank you for responding to that. We've got a few more questions for Dr. Stiber, and here's the next question. Please explain the overlap of your division with the Cder Drug Shortage Division.
Thanks for that question. It's a good opportunity to explain. I don't want to say overlap, but I want to say that certainly within my office of pharmaceutical quality, we work closely with the Cder drug shortage staff, and they are people that we're involved with in understanding what drug products are at risk of shortage, and that there's information that goes both directions. And so it's a very important interaction within our office. Thank you, Ray.
Thank you for responding to that question. We've got a few more questions for Dr. Stiber. Here's the next question. What does the FDA believe caused the spike in customer complaints in hand sanitizers in July 2020?
Well, that's a great question. I can't tell you necessarily what the FDA believes. I'm here just representing myself, but from my perspective, that spike in customer complaints was caused because there were a great number of new manufacturers of hand sanitizers, manufacturers who did not have prior experience with manufacturing Cder-regulated drug products, and some of these were non-traditional drug manufacturers. And so there was some time in which there were an increased number of complaints. A lot of that was also influenced by products coming from Mexico. And after the import alert for the hand sanitizers from Mexico, that helped to reduce the number of complaints. And also, as Connie has spoken about, the do not use or should not use list that FDA publishes online, that I think also helped to reduce the number of consumer complaints. So thank you, Ray, for that question.
Thank you for responding to that. We do have a couple more questions for Dr. Stiber, and here is the next question. If the sponsor has evidence of a contamination and the site is not addressing the matter, what are the options to report or consult with FDA?
Great question. So I think one way to report to FDA that's really available to anyone is through MedWatch. And that's something that we'd encourage consumers, sponsors, anyone who's aware of a drug problem could use the MedWatch portal and report concerns to FDA. Thank you, Ray.
Thank you for responding to that question. We're going to move back over to Dr. Ruzicka. And we have a follow-up question and regarding some dates. And here's the question. Dr. Ruzicka's answer about the hand sanitizer manufacturer end date for distribution, is that 31 March 2021 or 31 March 2022? If they're allowed to manufacture until 31 December 2021, then it would seem logical the distribution date would continue for about 3 months instead of March 2022, not 2021.
Thanks, Ray, for bringing up the follow-up question. And I do apologize. It is March 31st of 2022. With everything that's going on, I guess I'm stuck in 2021. But it is, the distribution should should cease at the end of March of 2022. I apologize for that. Thanks, Ray.
Thanks for that clarification follow-up, Dr. Ruzicka. We're going to move back over to Dr. Stiber. We have a few more questions coming in. And here's the first question we just received. How do you suggest that the public use the findings of the report on the state of pharmaceutical quality?
Well, that's a great question, Ray. And this is a report that we've been issuing for 3 years. And so we're really not exactly sure how the public is using it. But we think that, first of all, the public should gain from it a lot of assurance that the state of pharmaceutical quality is good, and that in general, the dose that they're getting and doses that they're getting subsequently are good quality doses of the drug products. So I think that that's one message they should take from it. And overall, I think people can ask questions to their doctors, to their insurance companies, pharmacies, organizations that purchase drugs, and better clarify the quality of the drugs that they're getting. So I think having an informed public is important, and this is sort of a step towards that, to provide more information to the public about the state of pharmaceutical quality, so we have an educated public, and also have assurance and comfort to the public that they can trust the quality of the products that they receive. So back to you, Ray.
Thank you for responding to that question. We have another question for Dr. Stiber. And here's the question. What new FDA programs will provide for improved drug quality in the future?
Oh, great question. Yesterday and today at this conference, there's been a lot of talk from different emerging FDA programs that are in development that I think are going to do a lot to improve pharmaceutical quality. One of them is the Emerging Technology Program, moving towards more advanced manufacturing, continuous manufacturing, has a lot of promise for improving pharmaceutical quality. Also has been spoken about is the NIP program, the New Inspection Protocol Project, which is a way of improving the processes for inspections, making them more consistent and driving towards some more quantitative outcomes for inspections. So that helps us internally and should help to improve pharmaceutical quality. And then lastly is the Quality Management Maturity Program that was spoken about yesterday. That's an opportunity for firms to demonstrate, not an opportunity that exists yet, but something that's emerging, it's under development, but we envision it as an opportunity for firms to demonstrate what they're doing in terms of their quality management that helps to influence the quality of their products. And we think that all of these things will be showing improvements to the quality of pharmaceutical products in the future. So thank you, Ray.
Thank you for responding to that question. We had one more question come in for Dr. Stiber, and here's the question. How can I suggest a topic for a future report on the state of pharmaceutical quality?
Well, thanks for that question. I think that, as has been said, this is the third year that we've issued the Rsvq report. And we are looking for new topics. We're looking for what would be relevant and interesting to the public and to the audience in general. And so if you have any suggestions, you can please send an email to an email address, which I guess will be put in the notes box. It's cder-opq-inquiries@fda.hhs.gov.
Well, I want to thank both of our presenters for the great presentations and for answering numerous questions that came in. We'll now enter into our lunch break time, returning at 12:30 p.m. Eastern Daylight Time. Please enjoy your lunch.